Hey, Sasha, can you hear me? Okay, I guess I have a problem with the sound. Can you hear me, Sasha?
Yes, I do.
Okay, good. I'm just checking. It's only you and me, but we will wait for a few more minutes and then. Okay, Jennifer. Hey, Jenny. Can you hear me, Jen?
Hi.
Hi, sweetie. I mean, I think I have a problem with the sound. That's why I'm checking if you guys can all hear me. Yes, so you can. Okay, good, 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 good. How's it going? Are you studying?
Yeah, just starting. I had to mark, I had all my labs at ITA. There's a scavenger hunt. They had to go find rocks. And so I had to mark write-ups on over 450 rocks.
Oh, goodness. I don't want even to think how much fun you had.
It probably took me about 30 hours.
But I would say that's good. I would say that's good. Yeah, you were pretty efficient. Yeah, yes.
Yeah.
But I mean, I would probably get tired. Like every time when I am marking you guys, I mean, you like generally, I get tired after like 3 hours, and then I just have to stop, or my brain simply doesn't function anymore. So
Yeah.
it's like I see all pretty colors. I saw your exam, so it so I think I succeed to get it a little easier because there are some things that I thought that are too hard for you. But when we go through the exam on Friday and Monday, I will try to exactly tell you on what to pay attention because exam is, I mean, it's not hard so much. I mean, you just have to know. That's the thing. You have to understand. That's all. And again, I will do my best to tell you what to study on. Yeah, give me just a second. I need to text one, just to send one message and then we are starting. So again, I will go through, I was thinking to go through units 1, 2, and 3 today, which is, I would say, a little easier than everything else. And you guys stop me if I'm going too fast, but again, I will just emphasize that we already went through all of this. Like when we were talking first time, this is just recap. So I hope it will be fine with you. Final exam doesn't have a lot of things from first part, which I don't like because I find first part easier. So, but I'm not surprised because real physiology is actually second part. So I will share my screen in a minute. And again, please stop me every time when you think I am too fast, because I know that I can be fast. Can you guys see presentation? Okay. So, again, stop me every time when I am too fast, if I'm too fast. So units 1, 2, and 3, in my opinion, pretty easy. Not for now, not many questions there, but again, there is no, so Lisa still didn't complete the exam because I gave some of my suggestions, so I'm still waiting for final decision. She might add something from these 3 units, so pay attention. But basically everything what I'm talking about here is what you need to understand. So So again, gram-positive and gram-negative cell wall. I see this as a basic of microbiology, not microbial physiology. So when you go out from this class, you need to know that gram-positive cell wall has thick peptidoglycan, teichoic acid, lipoteichoic acid, and some wall-associated proteins. And that's a thick cell wall, usually very hard to lyse. Gram-negative cell wall is the one that I personally like to deal with because it's easy to lyse, because it's thin peptidoglycan, it has some lipoproteins, LPS, which is the key, so pay attention on LPS. LPS is what you need to understand why it stands for, because if there is a question, it will be about LPS. Outer membrane, so again, that's slightly, it's a different structure than gram-positive cell wall. So I like to think about gram-negative cell wall as about sandwich, where peptidoglycan is in between two membranes. And of course, there is a periplasm. Archaeas, we had a few presentations about archaeas. They have specific cell wall, and I was impressed with presentations about archaeas because personally, I don't have much knowledge about archaeas. So archaeal cell wall is specific. It contains glycoproteins with various shapes. S-layer is how we call it. So if there is a question about these archaeas, it will be about S-layer. So that's what you should understand. And also, this peptidoglycan is not real peptidoglycan, it's pseudo-peptidoglycan. So it's not the same as bacterial. Yeah, so how bacteria are moving. So those are also parts of cellular structures, flagellum and archaeolum. Flagellum for bacteria, archaeolum from for archaeas, how it's synthesized and how it functions. So again, there is a specific controlled process of flagellum and archaeolum synthesis and function. They're slightly different. What is most important for you to memorize, and again, I won't read each of these because you can all read these things and I'm uploading that online, but what is important to understand for these two is that flagellum from bacterium is turned on with PMF or proton motive force, where archaeolum is turned on with ATP. So this is the basic, like nobody will ever ask you to write down like how synthesis of these things is going, but what you might be asked is how you turn on flagellum, how you turn on archaeolum, because those are two key differences. Type 4 pills, yeah, so again, similar like archaeolum in terms of ATP hydrolysis. So we had this question for midterm, and that one was designed by Lisa, not by me, to be honest. Many of you took that question, and I had a feeling that you understood how cellular synthesis goes. So what you need to understand from this part is what is happening with mutants PIL-Q, PIL-T, and PIL-Q and PIL-T, actually, yes. So if you have delta PIL-Q, how the mutant look, and if you have delta PIL-T, how mutant look. Again, nobody will ever ask you to explain in details how is this structure synthesized, but what I personally like to do is to write it down. When I understand how structure is synthesized, then I can understand what's happening if there is no particular component. So PIL-Q creates channel for PIL-in to pass, ATP hydrolysis pushes PIL-in through, and PIL-T degrades fiber, and this is how it moves. So it's retraction, so twitching motility. Yes, cell wall monomer biosynthesis. Again, if there is a question about cell wall, this will be a question. So how, what is the order of biosynthesis of cell wall? And you had that similar question on, I think it was first problem set, where people were struggling. I mean, personally, I was struggling first time when I saw that question. Like, I had to be very careful. So it's not easy question. So just understand basic points and you will have easy time. So here is how the whole process goes. So in cytosol, bacterial cell creates molecules that are needed to build cell wall. So everything is happening inside cytosol. So one of the dominant molecule of cell wall is NAG. So I will use name NAG because it's easier for me to pronounce, but the full name is N-acetylglucosamine. So I will use NAG just to be easy. Now, NAG is linked to UDP in the cell, and this is happening in the cell. So again, location of all these events is important. Then glycolytic molecule, PEP, is hydrolyzed, which now releases energy. This is good because now you know what PEP or phosphoenol pyruvate does. It's a high phosphate bond. It's a really high energy, and that's how you get energy that you need for all further purposes. So that energy is now used to attach, 3 extra carbon atoms to NAG, which result in NAM, N-A-M. So NAM is another monomer required for cell wall biosynthesis. So NAG and NAM. And again, everything is still occurring inside. Now, with NAG and NAM molecules being ready, cell wall biosynthesis can begin. So this was just preparation. And again, I circled here those two because they are key. Now, there is that conserved two-peptide structure. Diala is attaching to the terminus of peptide structure. Sorry, my bad. I should start from beginning. So now when you have NAG and NAM synthesized, UDP is attaching to NAM, and UDP NAM is attaching to 3 peptides that vary among species. Then you have that conserved motif that is attaching to terminus of peptide. This is now NAM pentapeptide. Again, we're still in the cell. All this structure is now transferred to bactoprenol phosphate or bactoprenol, you can call it as you like. Now UDP will bring NAG and attach it to NAM pentapeptide bactoprenol. Molecules that are called autolysins will basically make a space, open cell wall, so they make a space for insertion of new cell wall monomers. Now bactoprenol is still attached to NAG-NAM pentapeptide, and it flips it over to the periplasmic space. What's happening here now? So you have now structure on the other side. So just flip. And now you have those transglycosylases that are forming bonds between NAG and NAM in periplasm. Now we are out of cell. So bactoprenol is just translocating it. And then transpeptidase forms bonds or bridges between 2 diala motifs in periplasm. So this is basically what is important for cell wall monomers to know is where things are happening. And of course, what's the order? But I think that I would pay more attention on location, like which, like for example, transglycosylases and transpeptidase are operating outside in periplasm. Now, penicillin antibiotic resistance. Again, I don't think you will have a question about that because this is common knowledge, but I'm just going to run through that fast. So for general purposes, like it's cool to explain if people ask you, because penicillin is a worldwide famous antibiotic. So how it works, it mimics diala motif, and now the transpeptidase cannot bind. So it's like a tricking this enzyme. So vancomycin, similar way of mode operation. It recognizes and masks diala, and now transpeptidase cannot bind. So similar, but not the same. But again, I don't think it will be these type of questions. So antibiotic resistance was on midterm. I don't think it will be in this exam. Cellular division. So I will tell you what to pay attention in this physiological basis of cellular division. There are some important parts that I think Lisa wants you guys to know. So there are those 2 proteins, min proteins, E and D, that we were talking about, and they're expressed together. So D leads E. So D basically helps min E oscillations. When chromosome is completely replicated, that's a sign, that's a signal for min proteins to stop oscillating. Now cells have 2 fully formed chromosomes, and now everything else can divide. Next one is min E ring formation. And again, what I think it's important for you to memorize is who is leading oscillations and who is forming a ring. So min E is forming a ring. And FTSZ, which is a key protein, replaces min E. So I believe that these are most important things to memorize. Like if I ask you which protein in cellular division is the key master regulator, you would probably say FTSZ, because not min E, but it wouldn't be completely incorrect if you say min E, but I don't think you would get points. I mean, I would probably give you, but I don't know if Dr. Stein would, so pay attention to that. Next one is ZIP-A that anchors FTSZ. And FTSZ is doing ATP or GTP hydrolysis. So again, FTSZ is what I would memorize, like all main characteristics, because that's a key regulator of physiological basis of cellular division. Then ring constricts, like now division can be completed. And we have these autolysins that create space for new cell wall polymers. Again, autolysins are, again, molecules that I would memorize because they are important. They are pretty much key point in forming cellular structure of bacteria. They're opening a cell wall, they're making a space for new polymers. Without them, you can't get cell division completed. So memorize autolysins. Fatty acids. This one, I think it was something similar on midterm. So I think that there will be a question about fatty acid biosynthesis, and you do need to understand exactly how this process functions and which molecules are important for fatty acid biosynthesis and the order. Again, you have access to your notes, but make sure that when you have notes, you create those notes so you can easily find things. Because this exam is time limited, and if you start digging and searching around, you will lose time. So the key point, acetyl-CoA. Now we know about acetyl-coenzyme A. This is basically mastermind of cell. So carboxylation of this acetyl-coenzyme A gives malonyl coenzyme A. And you don't have to memorize this large enzyme ACC, ABCD. What you need to know is that this is carboxylation. So adding carbonyl group. Now, when there is malonyl coenzyme A, this malonyl coenzyme A is transferred to ACP. ACP, acyl carrier protein. Again, very significant compound. The enzyme that is basically grabbing malonyl coenzyme A and transferring to ACP is FAB-D, labeled here in red. Now, next step is condensation of this structure, malonyl coenzyme A attached now to ACP, and acetyl moiety. So basically, malonyl and acetyl needs to be condensed. Just malonyl is attached to ACP. And the enzyme that does this is FAB-H. And now you get, as a result of this condensation, beta-ketoacyl ACP. So you had carboxylation, you had condensation. Next one is reduction of this all ketoacyl structure to hydroxyacyl ACP. And the enzyme that does that is FAB-G. So again, you had carboxylation, condensation, reduction. Next one is dehydration, which gives you trans-2-enoyl, and enzyme that does it is FAB-A. And now is the last one, again, reduction via FAB-I, and you have acyl ACP. So we say this is carboxylation, dehydration, reduction. But before, on the top of everything, you have condensation. So I think that as long as you understand principles, it won't be a problem, even if you don't memorize all these names. I mean, try to memorize, but again, once when you understand how process function, like you have condensation, actually carboxylation, condensation, reduction, dehydration, and then again reduction, it should be fine. And again, acyl ACP will be used for biosynthesis and phospholipids. This was one of key issues on midterm. Is it, which molecule is the key? Is it DHAP or glyceraldehyde 3-phosphate? And again, I was accepting both, but the key molecule is dihydroxyacetone phosphate. This one comes from glycolysis. And then again, DHAP is reduced via glycerol 3-phosphate synthase. This is called glycerol SN3P. Then enzymes, Fab enzymes, HGA, are creating fatty acids. So we already went through that. So fatty acids are formed, glycerol is formed via glycerol synthase, glycerol phosphate, and now you connect them together. And that's done by acyl transferase enzymes. And the result is phospholipid. So basically think about this process as like from the two parts: glycerol phosphate, fatty acids, condensation via acyl transferases. Oops, sorry. So the same how we had penicillin and vancomycin. So penicillin and vancomycin are antibiotics that are intervening with cell wall biosynthesis, mostly with transpeptidase, not mostly, with transpeptidase. Other antibiotics like triclosan are intervening with fatty acid biosynthesis. And again, this antibiotic is binding Fab-I, final enzyme, and that increases affinity for oxidized form of NAD. So affinity for NAD plus increases, and that means that process cannot be continued. Isoniazid, again, like target is the same, Fab-I, just a slightly different mechanism. So the mechanism of this antibiotic is creation of isonicotinic acyl radicals, and interaction with NADH gives this nicotinoil something NAD, which binds Fab-I. But what is important to understand is that Fab-I, the final enzyme in fatty acid biosynthesis, is the target. So I don't think again, I still don't think that you will have question about antibiotic resistance, but this is just general like culture. Platensimycin is not, so this one is affecting Fab-H. And that's all what I would memorize. So all of these antibiotics are affecting Fab enzymes. Triclosan, isoniazid, Fab-I, where platensimycin affects Fab-H. So membrane-associated transport. Oh my God, this was so complicated for me. I really struggled to organize it nicely. So when we think about membrane-associated transport, we think about proteins that are on cell membrane and that are mediating transport of various compounds through the cell. So first one that I have circled here are channel proteins. Then we have carrier proteins. They are secondary transporters, which means they are coupled with PMF. So when we say channel protein, the way how I understand it is basically it's one protein embedded in membrane, and that protein has a pore in it, like a channel, and small ions are just traveling through it, depending on concentration gradient or the cellular need. But usually process is way more complicated. Carrier proteins are proteins that are PMF coupled. That means that in order to transfer something across the membrane, these proteins have to use PMF, proton motive force. And there are carrier proteins that use ATP to transport something across membrane. So what are channel proteins? Usually transport very small solutes, ions, different charges and sizes. And I have an example here about mechanosensitive channel. So again, this is what I found in literature. To me, it was interesting to know. But this is how bacteria regulate their hyposmotic environment. So let's say that this is one of extremophilic adaptations. And again, I won't go through details how this functions because this is something that I just found. It's not even in lecture. But for me, it was interesting to understand the channel function itself. Secondary transporters. So what is the main characteristic of these transporters? Conformational change in response to solute binding. So something binds. Protein is changing its conformation and the solute is released from the other side. So usually it's like a gate. So it binds, protein changes conformation, solute is trapped, travels through the receptor, gate opens on the other side, and solute comes in the cell. So again, this was my just brief explanation. So in closed state, there is a solute binding domain located outside, specific for specific solute. When solute binds, carrier is going through conformational changes, and then you see solute trapped in the carrier. When carrier is in open state, solute binding domain is formed on the inside. So basically, that solute binding domain is like moving through the receptor, and then solute is delivered through cytoplasm. So I think about this transport as about like a water bed, like as you are moving, that's how it changes the shape. What's this? Trap transporter. Again, all what you should memorize here is that this is sugar transporter. So what is transporting and how it's turned on. So I think this is mostly for glucose, or probably there are other sugars that are released. So what you need to know is what turns it on. So proton motive force and sugars are the one that are transferred. Symporter, lactose permease. So how this thing functions. A little confusing, really, for me, so many different transports. Again, this is just a different example of transport. So LAC-Y is in outward open conformation. So again, ready to accept substrate. H plus or protons are binding to this transporter. And this is followed by lactose binding. So lactose is the one that you want to transfer inside the cell through this LAC-Y. So when lactose is bound, which is helped by protons, there is that conformational change of receptor, and now all receptor is taking inward conformation, like open conformation. Lactose is released, and protons are also removed because you need to release it. So now LAC-Y is ready to get proton again. So basically, just a base mechanism of transport is important to understand. Binding of the substrate, changing conformation, changing of conformation, transfer through the receptor or transporter, deliver of cargo into the cell. And that can be helped by, in this case, proton motive force. So this was symporter, which means symporter means that two solutes go in the same direction. So in previous case, that was protons and lactose. Antiporter means one thing goes out, the other thing goes in. So just exchange. Export of drugs from bacterial cell, very, very often a mechanism of bacterial resistance on various toxins. So but it's pretty straightforward. So when bacteria runs into some toxic drugs, it wants to remove it from cell. So those antiporters will do that. The first step of this is binding H+ at periplasmic site of the membrane. So H+ is bound, and next step is drug binding from cytoplasmic side of the membrane, because drug is already inside. When bacteria detects drug inside, that's a signal that the drug has to go out. Protons are binding from the top, drug is binding from the inside. That's why it's antiporter. It goes in two opposite directions. Now, when drug binds, again, the same principle, conformational change induced by both drug and H+, bound from different sides of membrane. Next step, the drug is released to periplasm and H+ leaves the structure. It goes out from antiporter. Now antiporter is ready to pick up another molecule or drug inside the cell. So basically, again, the same binding of target, conformational change, export or import. So one more example, pretty much similar. So I won't go into the details how this go, because you can always read this. I think this is pretty clear. So again, binding a substrate, changing conformation, and delivery of cargo, export or import. I don't think there will be any question about these transporters, at least not in the version of exam that I saw. But you should definitely be aware of that, and you should memorize these things because it might be needed to answer some other questions. Carrier proteins coupled to the energy source. So ATP or other. What can be other? Phosphoenolpyruvate, for example. So phosphotransporters and ABC transporters. The most famous one are ABC. So if there is a question about transporters, I'm pretty sure it will be ABC transporters because these are the most famous. PTS transporter is glucose binding transporter. This is interesting mechanism, and you might want to understand the process of binding through these enzymes, actually these structures. I would pay attention to this because, like, even though I believe that ABC are more popular and more significant, this is interesting process. So again, try to understand. So phosphoenolpyruvate hydrolysis is the one that gives the phosphate group that attached to EI part of transporter. So EI is just the protein part of the transporter. Phosphate group is then transferred from EI to HPR, and then from HPR to E2. So you have EI, HPR, E2, and then E2A accepts phosphate group and transfers to E2B. So you have various of these phosphate transfers between different proteins, different complexes of proteins. But finally is that phosphate is added to the glucose, and glucose is attached to E2C. So E2C is protein that holds glucose and waits for phosphate to come through all these proteins, E8, 2, blah, blah, blah. So glucose waits on the membrane. Now, when you have glucose 6-phosphate, that's activated molecule, and that goes through glycolysis. Glucose 6-phosphate, now you know glycolysis, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, or it can go through EDD pathway depending on bacterium. But glucose 6-phosphate is what you need to have so you can complete all cellular biosynthesis. ABC, famous one. Again, mostly for drug export. So bacteria doesn't want drug in organism. To export it, it's using ABC transporter. So drug binds to the domain located inside at cytoplasmic site of the membrane. And now you have ATP molecule in the next step. So hydrolysis of ATP is the one that is needed to turn on this transporter. So again, transporter is changing conformation. Drug is now trapped. When ATP is hydrolyzed, drug is released to periplasmic side of the membrane. So all these processes of ATP binding and drug binding are happening simultaneously, but when hydrolysis happens, that's like a push. Yeah. Again, two-component system. Please understand this. Doesn't matter how the protein is called. What matters is all process. Like you have various different proteins, various different processes that are controlled with this two-component system. I don't think the name matters. What matters is to understand basic principle. And that basic principle is given here. There is a structure, sensory kinase. So it's a protein that senses what's happening in the environment. The first step is the drug binds to that periplasmic domain of sensory kinase. Sensory kinase is something that phosphorylates. Kinase is enzyme that has phosphotransfer activity. It's doing phosphorylation. So when drug binds, this phosphorylation activity is turned on. And what's happening is activation of sensory domain of sensor histidine kinase. So periplasmic binding, activation of cytoplasmic sensory domain. Now that kinase is bending and phosphorylating itself at histidine. That's why it's called histidine kinase, because phosphate group is added to histidine. Now, in that phosphorylated stage, which is called autophosphorylation, this sensory kinase can complete further phosphorylation of downstream response regulator. So downstream response regulated is phosphorylated at aspartate residue. Phosphorylated response regulator will now be able to go downstream further and to activate transcription of some genes. Usually that response regulator can be transcription factor or can activate some other transcription factor, but this is the basic principle of two-component regulatory system. And that's all for units 1, 2, and 3. So how do you feel? Is it hard?
Thank you so much for doing this. It helps a lot.
I mean, I think it will be the most helpful thing will be on Friday when I actually lead you through exam. But I think this is useful because I am refreshing your memories and I am refreshing my memory as well, because I have a hard time memorizing things. I mean, oh, Brittany, hi, I didn't see you. So, I mean, we will meet again tomorrow and go through units 4 and 5. I think that exam will contain both 4 and 5. Wait a second, Dana asked, will the Friday and the next Monday sessions be the same? No. Friday and next Monday will be basically only the exam. So now I am going through each of these, basically like a recap of all class very fast. And on Friday and Monday, no, but it will be the same Friday and Monday, if that's what you are asking. Yes, the same will be on Friday and the same session will be given on Monday for those people who were not able to attend on Friday. So in case you have other things to do, which I'm sure you have, I will give exactly the same sessions on Friday and Monday. And oh, Brittany has a cat. Here is a kitty. Yeah. Oh. Sorry, guys, I love cats really much. So every time when I see cat, I'm like, oh, cat. Sorry, Dana, I didn't first get your question. So yeah. Anyway, that's it. So on Friday, I should have an exam. I saw first draft of the exam and I had some suggestions. So Dr. Stein incorporated them, thank God, luckily for you. So will be good. but I still didn't get final version. So I'm hoping I will get it by Tuesday. That's how she told me. So tomorrow we will work on other things on 4 and 5.
Is the exam that we're looking at on Friday and Monday like from a past year?
No, from this year.
It's the actual exam? Oh my God.
I mean, I do have access to that. So I find that very useful for students because I think that you guys cannot know what to study and how to study if we don't tell you. You might end up, if we don't have those type of sessions where we actually go through the exam, you might pay a lot of attention to some meaningless things in lecture and you miss some important stuff. That's why I insist on giving this type of reviews where I actually see the exam. So I can direct you how to study. I cannot tell you, of course I can, but I can tell you what kind of question will be, so to tell you what to memorize, what to understand. I mean, I personally had issues with that type of exams where I don't know how questions will be asked. So Laiba asks, for a second, I thought you meant the actual exam we are going to be taking. Yes, yeah, it will be actual exam. Like Dr. Stein sends me an exam, I see the exam, and I write review session based like a question 1. Pay attention to this, this, and that. Question 2, pay attention to this, this, and that. That's how review session goes. Yes, that's the thing. You guys have to understand how we ask questions. If you might end up knowing so many things, but you just don't know what we will ask you, and then you get lost in so many information, and then you mess up, but you actually know. And I would feel really bad if that happens. So let's make sure that doesn't happen.
Yeah, that's exactly what I've been worried about, just because there's so much content, like going through, there's like over 70 slides in each unit. And so it's a lot of content. So thank you so much for helping distill it down.
I really want you guys to do well, because if we don't work with you, that's like, I think that if students don't do well, that's like 50%, if not even 80% professors and TAs responsibility. So I usually say TAs because I am always TA, I'm still not professor. So if since my job is to assist in teaching, if I don't assist well and I don't teach you well, that means I didn't do my job, and I feel really bad, and I want to do my job well. So we will go through each of these things separately. And then on Friday and Monday, we go through full exam. So attend. Last term, I did the same thing, but nobody came to review session, and then they all struggled and they all messed up. So
I can't believe there's only 12 people here for this even.
I agree.
Why wouldn't you come for this?
It's
technically it's only 11 because Marina's the TA.
Oh, that's true.
Probably because it's recorded though. Because like I know for undergrads, like I have 4 exams right now, right? And like today I have 2 quizzes and 2 assignments due. It just so happens that I can still make this, right? Because it's recorded, people probably will watch it later.
I mean, I hope it's
still classes too. Like I had lectures and I still have lectures after this. So I think a lot of people are like in their lectures today.
Okay. I mean, I hope it's not because people hate me. I mean, I really hope it's not. I don't. I mean, who knows? I mean, I can be bitchy. I know that. Ask my husband.
I've never had it.
Oh, sorry.
No, go ahead, Brittany.
Oh, I feel like a lot of people though, because the exam is until December 17th, that they sit back and they're like, the exam's in 10 days. I have a final on the 11th or the 14th. So they're just focusing.
And then it's so nice that this is recorded, right? Because like people who have finals this week, who have finals like Monday, Tuesday next week, can still like attend these sessions just like after the fact.
Oh, I will be. And here is the thing, guys. I am uploading everything. Like this presentation is uploaded under review material. But as presentations from review unit 6, 7 up to 10 is not, because I finished it on Friday. So I will upload everything and you will have all of that. Like I know that some my colleagues are downloading these materials because everything is synthesized in one document, and I'm going to use that for my candidacy also, because I'm pretty sure that where is Carmen? Yeah, Carmen and I have Lisa in committee, and yeah, it will be fun.
I'm in denial.
No, Jen, is Lisa in your committee as well?
No, thank God.
Well, here's the thing. She's much better than some other people. Like, I actually heard that she belongs to the best professors for committee. Like, you would be surprised what kind of people are out there. I mean, I'm sure graduates wouldn't be surprised, but undergrads would be. I cannot be surprised with anything else. I think I saw all. So anyway, I don't know how to upload this to the e-class. I think that this goes to cloud now, and I will get an email, and then I have to convert it to the YouTube. So I'm going to talk to Lisa to help me with that because she knows how to do it. So I will text her probably in a few minutes, like just to meet with her later today so she can explain me. But you will have, we are meeting tomorrow, so we have time, okay?
There's also a link like on the e-class page where I joined this meeting. It says like upcoming meetings, previous meetings, and then cloud recordings. So, you can probably upload it to YouTube, but it looks like you can also upload it to like this thing right here that says cloud recordings.
I think the same. I think that's why I clicked cloud, upload to cloud, but I don't know how it functions.
Yeah.
Lisa was mentioning that I should convert it to YouTube just to avoid overwhelming e-class. So I will find out more later today.
Thank you.
I'm really stupid for technology, and this is for me pretty new period. Like when everything is online. I'm a person who still prints everything, so for me this is like I'm living on Mars or something like that. I am doing everything online, so I will learn slowly.
Yeah, no, this is great. Might not make the first half of the Friday session, so I'm glad that you're recording everything.
Come on Monday. If you're free on Monday, come on Monday. And again, I'm very willing. If I have time, I will meet with you. Now I start working on some very big experiment that's really taking me a lot of time. So I'm literally sleeping in this place. And I have part-time job, meaning that I have to drive again over there. So if I don't have time to meet separately with you, that's just because I'm too busy, not because I don't want. Anyway, I am leaving to deal with proteins. See you tomorrow at 11. Okay?
Thank you.
Thanks, Maria.
See you guys. Bye.
Bye.